So we're going to wrap up our nucleotide nucleic acids today by looking specifically at the way that DNA behaves. So we talked briefly about the structure of DNA and RNA. So we reflected on the fact that DNA was more stable than RNA, and I wanted to go back and recap that a little bit. So RNA is unstable under alkaline conditions. You can have hydrolysis that's catalyzed by enzymes like RNase, and they're abundant. You have RNase in plants to prevent inbreeding. You have RNase P, which is a ribozyme that processes the tRNA precursors. And dicer is an enzyme that cleaves double-stranded RNA into oligonucleotides. And again, these are both protection from viral genomes as well as can be incorporated to be used in RNA interference technology. So if you look at the mechanism of base-catalyzed RNA hydrolysis, what you actually see is the abstraction of a proton, then the attack of the electrons from that bond onto a phosphate. You see the cyclic formation of the phosphate, and then this shortened RNA. Because two bases can hydrogen bond to form a base pair, for monomers, a large number of base pairs is possible. However, in polynucleotides, there are only a few possibilities that exist because of space and directionality. So the Watson-Crick base pairs predominate in double-stranded DNA, where A pairs with T, C pairs with G. Ideally, that's a purine pairing with a pyrimidine. We can actually see the distance in terms of the bond lengths and the way that these H bonds line up with each other at the respective heteroatom hydrogen pairing. Because the discovery of DNA was so pivotal, we want to take a couple of minutes and just recognize why this discovery was important. So Watson and Crick reported this in Nature in 1953, but we know that their discovery was really built on the work of many other individuals like Linus Pauling, Rosalind Franklin, and Maurice Wilkins. And it's a great illustration of the way that sometimes the misconception of knowledge as well as collaboration and sometimes the unfortunate consequence of not having a full understanding of data. So in 1929, Levine and London proposed a structure of DNA and then modified that in 1935. And these, again, were the result of both hydrolysis and chemical analysis. The Watson and Crick model was the first to propose the double helix model. Linus Pauling had proposed a triple helix. There are three forms of DNA that are most prominent. There's the A form, the B form, and the Z form. And so we want to think about a couple of things that influence this form. So specifically, we want to think about the forces that determine DNA structure. So those are going to be electrostatic forces or repulsion forces, hydrophobic forces, H bonding, and then stacking interactions. So the hydrophobic effect is where base pairs are the interior of the helix. They're hidden from water. That means that they're charged and hydrophilic. So you have your phosphate backbone on the exterior. Then we have these stacking interactions, which is the close contact of the base pairs, where the plane is perpendicular to the helical axis. We have pi-pi stacking, van der Waals, and dipole-dipole interactions that stabilize the double helix. And that's as much stability as a pair of H bonds. We also have these ionic interactions or repulsion, where these forces destabilize the double helix. The phosphates from the two strands are kept away from each other to reduce electrostatic repulsion. So at normal pH, oxygen is negatively charged. And one of the things that we see are the H bonding between the base pairs. So 2 H bonds for each AT, 3 H bonds for each GC. And we see that these hydrophobic effect and stacking interactions are not sufficient to overcome the electrostatic repulsion of the phosphates. So H bonding is really what guarantees the complementarity of the strands. So if I look at the three forms of DNA that have been solved, I see that the A form is the dehydrated form, and that the B form is the hydrated form. Most DNA looks like this. And then the Z is the zigzag form. This is long special sequences that can adopt this conformation. So because the B form is dominant form in nature, we want to take a little bit more of a look. So we have 10 residues per turn of helix. So one turn is 4 angstroms, right? Or 34 angstroms in length for a whole turn. These base pairs are almost parallel to the helical axis, and then the sugar pucker is C2 endo. All bases are going to be anti, and we have a major and a minor groove. If we compare that to the A form, The A form does not exist to a significant extent. There are 11 residues per turn of a helix. The base pairs are highly tilted, and the sugar pucker is C3 endo. The bases are all anti. Major and minor groove due to orientation of the sugars with regard to the base pair, and it's somewhat relevant for double-stranded DNA in terms of just an understanding of the contrast. So alternate structures relieve stress. The Z form of DNA can relieve negative supercoiling by forming short regions of left-handed helix. And then we also see some other forms we'll talk about in a bit. So the complementarity of the DNA strands, two chains are going to differ in sequence. So you read from 5' to 3'. So these two chains are complementary and they run anti-parallel. Notice the 5 to 3 and then the 3 to 5. So if I want to look at how to replicate my genetic code, the first thing that's going to occur is strand separation. Each strand serves as a template for the synthesis of a new strand, and synthesis is catalyzed by enzymes known as DNA polymerases. So this newly made DNA molecule has one daughter strand and one parent strand. This is what's known as semi-conservative replication. And you can actually see that here. This messenger RNA gets synthesized as the carrier for the information of what determines the sequence of proteins. So it's synthesized using the DNA template. It contains ribose instead of deoxyribose, and it contains uracil instead of thymine. So one mRNA can encode for more than one protein. Together with transfer RNA, this is what transfers the genetic information from DNA to proteins. So if we think about the fact that monocistronic, right, that's a single gene, as opposed to polycistronic, that's multiple genes being carried on a sequence of DNA. One of the things that we can see here is this is our RNA, as opposed to our DNA, where the DNA is blue and the RNA is green, just as a comparison. So palindromic sequences can form hairpins and cruciforms. So palindromes are words or phrases that are the same when read backwards or forward. If I look at a palindrome, right, that's also just like a mirror repeat. And one of the things that's really interesting about this is that I can actually form then specific sequences in DNA either into hairpins, okay, or RNA molecules that have very complicated structures because of these hairpins and cruciforms. Notice some of the coiling or turns that I can see. Now it's important for us to talk here about DNA denaturation. So this is where covalent bonds remain intact and the genetic code remains intact, but the hydrogen bonds are broken. So the two strands separate, the base stacking gets lost, and the UV absorbance increases. So denaturation can be induced by high temperature or some type of change in pH. And this denaturation can in fact be reversible through a process that's known as annealing. So here's our double helical DNA, can undergo a denaturation process, right, and an annealing process to take it back to the original double helical form. Thermal DNA denaturation is what's known as melting. So this is where the DNA exists as a double helix at normal temperatures. The two strands then dissociate at elevated temperatures, and then the two strands re-anneal when the temperature is lowered. The reversible thermal denaturation and annealing forms the basis for the polymerase chain reaction, which we're going to study in just a little bit. And this DNA denaturation is commonly monitored by UV spectrophotometry at 260 nanometers. So you can actually see here the melt time, or melt temperature rather, and its relationship to denaturation. The factors that affect DNA denaturation is the middle point of melting, that's the TM, that depends on the base composition. So if I have high areas of CG, that's going to increase my TM. TM also depends on DNA length, so the longer DNA has a higher TM, and it's important for short DNA too. TM depends on pH and ionic strength, so a high salt increases the TM. You can actually see here the percent of total nucleotides with respect to their TM and the way that they cluster. So if I look at the denaturation of large DNA molecules, it's not going to be uniform. So at rich regions, there is a lower melt temperature than in GC-rich regions. If I wanted to detect specific DNA molecules in a complex mixture, I could use radioactive detection, fluorescent DNA chips. I could also amplify specific DNA and look at the PCR, or site-directed mutagenesis, And I would be able to study the evolutionary relationships for the antisense therapy. So let's say I had 2 samples, I could mix and cool them. I would see a hybrid sample, a duplex of sample 1, and a duplex of sample 2. And if I wanted, say, for example, to look at spontaneous mutagenesis, I would be able to do several different things. I could look at deamination. I could look at depurination. And what's interesting is that most cells have mechanisms to correct for these. So deamination you can see here. Depurination you can see here. I also, though, see oxidative damage. Mitochondrial DNA is most susceptible to oxidative damage. I also see chemical alkylation. And again, cells have an ability to correct some of these changes. So here, alkylating agents. UV light induces dimerization of pyrimidines. This can be the main mechanism for some types of skin cancer. Ionizing radiation causes ring opening and strand breaking. These are particularly difficult to fix. Cells can repair some of these modifications, but others cause mutations, and accumulation of mutations is linked to aging and carcinogenesis. So you can see here an example of damage and a kink. And then there are other functions of nucleotides as energy sources. So, for example, sorry. Oh, why is it doing that? Sorry, let me fix this. For example, as energy sources, we actually see AMP, GMP, CMP, and UMP as the monos, ADP, GDP, CDP, UDP is the di, and then ATP, GTP, CTP, UTP as the tri. And these are other functions. So this in this case is energy storage. And then if we look at coenzymes, it should be no surprise that I also have nucleotides that can function as coenzymes, where the 3-phosphoadenosine diphosphate, or the flavin adenine dinucleotide and nicotinamide adenine dinucleotide are the basis for coenzyme A, and these other electron transport molecules. And finally, we also see regulatory molecules. So these are molecules that play a significant role in cell signaling. You can take a look at their structure here. And that's it. So we've covered a significant amount of information. So one of the things that I want you to be thinking about is the extent to which both our RNA and our DNA are pivotal when it comes to both storing information, utilizing information to make proteins, and then, of course, converting those structures into cellular processes. So we'll talk about PCR in greater depth in class. Thanks. Have a great day.